let's do a quick review of microbial genetics. Remember, we followed in class the central dogma, so that means the flow of information. How in all living organisms that starts with DNA, and then we make a messenger RNA transcript of that, and then the messenger RNA is made into proteins. We call that process translation. Okay, so replication, DNA making more DNA from a DNA template, transcription making RNA from DNA. Okay, so replication doesn't necessarily need to take place first. In bacteria, we say replication begins at the origin. Okay, so all of the DNA gets made. We didn't really talk about an initiation and a termination so much, but there's a starting place, and that's called the origin. So let's see. If we think about all the different enzymes that are involved here, here's the old DNA, and you can assume that this has become unzipped here due to the enzyme DNA helicase. Okay, that's, again, we say unzips or forces those two strands of DNA apart. There's going to be some stabilizing proteins here that kind of keep that replication fork open. And then we have a leading strand and a lagging strand. No matter whether you're starting with the leading strand or the lagging strand, there's going to have to be a template, because DNA polymerase III, the main enzyme that's adding the nucleotides, the deoxyribonucleotides, they need to start with an RNA primer. Okay, so remember the enzyme that adds that, and we call that primase. Okay, and that's just going to be temporary because that's just a little short strand of 10 base pairs or so of RNA. And then the enzyme that I just mentioned, DNA polymerase III, okay, it will be able to start there from that RNA polymerase and proceed towards the replication fork. We call that the leading strand. Remember the directionality. It's always going to be adding on to, let's see, let's pretend, this is kind of weird the way I drew this, just a second here. Let me erase that. Here's the RNA primer there. Okay, here's the enzyme, DNA polymerase III. It's always going to be adding on to that 3' hydroxyl group. So here's the new strand. You know that wherever the DNA polymerase is, that's the 3' end. So this must be the 5' end of that new strand. So then remember, it's anti-parallel, so this is the 3' end of the old strand. Okay, so always adding on that 3' hydroxyl group. Okay, and we say it's leading because it's heading towards the replication fork. Then the lagging strand involves a series of primers. Remember, the RNA polymerase has the same directionality. So if this is the 3' strand of the old there, then this is the 5' strand down here. So this is going to be, we're going to have a series of primers here, and then the RNA polymerase is going to be moving out this way, and this way, and this way. Remember, we call those Okazaki fragments. Okay, like this, like this, like this. Okay, so there's a series of primers that the RNA polymerase starts from, and then remember those other enzymes, DNA polymerase I, they are going to get rid of the primer, because remember, it's RNA, and fill in nucleotides that match, that are complementary to the template strand there. And then the other important enzyme, DNA ligase, that seals gaps. Okay, so let's see, maybe I can change colors here. That the DNA polymerase will go back in. Actually, it's where the primers would be. Fills that in, and then there's going to be some ligase that will just kind of, it's just a phosphate group that has to be added there. So eventually, you have a continuous strand, of course, of DNA. Let's see, one other enzyme that we talked about that's important here besides DNA polymerase 3 and primase and polymerase 1 and ligase, there was an enzyme that worked upstream of the replication fork. And remember, that was called DNA gyrase. And that was important to relax the supercoils. So it makes cuts, spins it around, and seals it back up again. Otherwise, it would be too difficult eventually to keep opening up that replication fork. All right, so that in a nutshell is replication. Every time a cell divides, it has to replicate its DNA. Much more often, that DNA has to be used to make some kind of RNA. It can be messenger RNA, it can be ribosomal RNA, it can be transfer RNA. Okay, so remember, replication doesn't have to take place every time before you have transcription. It's constantly being transcribed. Okay, so during transcription, we'll hit the highlights here. If here is the DNA, it's going to be transcribed. There's an initiation stage that we talked about. The main enzyme that's going to transcribe the DNA is called RNA polymerase. And the place where it binds on the DNA, remember we had a special name for that, a special region. We called that the promoter region. Sorry, try that again. Okay, good enough. Okay, so that's upstream. Where it's going to start transcribing, maybe say, is right here. So the RNA polymerase binds to the promoter. The more tightly it binds, the more likely it is to actually transcribe the gene. So we talked about some things that make it bind, that make it recognize that promoter the best. There are something called, I don't really know how to draw this, I'll draw it as a little circle. Something called sigma factors that will bind to the RNA polymerase, again, that help it recognize the promoter and bind more tightly to the promoter region of the DNA. And then that sigma factors fall off once it starts transcribing the gene. The other regions that we said were important, remember there was a couple regions on the DNA, that -10 base pair region, and then there was I think it was -24 base pair region. Those had consensus sequences. So we said that the RNA polymerase especially will recognize those regions, and if that sequence of base pairs, again in that -10 and -24 region, is close to this consensus sequence, then the RNA polymerase is more likely to bind. Okay, and then elongation, we didn't really talk about that. So that's just when the RNA polymerase, again, is making a strand of RNA with that DNA template. And then in termination, it can happen by two different ways. It can involve a Rho protein, which is essentially a helicase enzyme that basically moves down the new transcript and dissociates it from the DNA and dissociates the RNA polymerase to end it. Or it could happen by a sequence dependent mechanism where we get those hairpin loops that causes the mechanical stress that causes the RNA polymerase to dissociate and the new transcript to come off. All right, so assume that that messenger RNA that was that RNA was made was messenger RNA. What happens to that? Well, in the process of translation, that gets read at the ribosomes. So this is just going to be kind of a basic reminder here. I feel like I can't draw this anywhere close to do it justice. But remember, the messenger RNA gets read at the ribosomes. So let's see. Let's draw this. Here's the small subunit of the ribosome. So remember, for bacteria, that's going to be the 30S size. Okay, eukaryotes have a 40S small subunit and 60S large subunit for a total of 80S. But bacterial ribosomes, 30S small subunit and 50S large subunit. Okay, so here's the messenger RNA. We talked about how it has to kind of dock in just the right place at that small subunit. We talked about a special sequence called the Shine-Dalgarno sequence on the messenger RNA that lines up just right with a certain spot of the ribosome so that the first tRNA that's carrying that's charged with an amino acid, the first one is always going to be formylmethionine, F-met, will come in and dock at the P site on the small subunit of that ribosome. So again, the start codon has to be lined up just right there. Okay, and that's the initiation of translation. And all the other new tRNAs carrying an amino acid are going to enter in at the acceptor site or A site. Okay, and so then you're going to have kind of as the messenger RNA moves through here, you'll have the new growing peptide growing from that P site. And after initiation, then the large subunit forms there a ribosomal complex. Forgot to draw that in. Okay, so that's kind of the gist of it, how it starts. How does translation terminate? Remember, when a stop codon is read, there's 3 different stop codons that every 3 base pairs, remember, makes a codon. And when a stop codon is read, then there's a pause in translation, and release factors come in and cause the ribosomal complex to dissociate, and the new end translation is terminated. So again, that's just an overview as you're studying for the final. Hopefully that will kind of jog your memory a little bit, and if there's details that are foggy, certainly refer back to your lecture notes or back to the book.